Hello everyone, welcome to one more interesting session of Biomolecule NCERT Revision. In this part, we are going to study about nucleic acid. So, general characteristic of the nucleic acid. Nucleic acid was first isolated by the Swiss biochemist Friedrich. Measured in 1868 from the pus cell and it named is nuclein. The term nucleic acid was first introduced by R. Altman. So these are some of the important dateline in the discovery of the nucleic acids. So nucleic acids are the third most abundant component after water and protein in the cells. This is the third most abundant biomolecule. Deoxyribonucleic acid found mainly in nucleus, chloroplast, mitochondria in all eukaryotic cells. So, if you remember, bacterial cell are prokaryotic. They do not have any nucleus. But this nucleic acid is present in cytoplasm itself. And it is called as nucleoid. It is called as nucleoid. So instead of nucleus in prokaryotic, nucleoid is present. But the DNA is there. Okay, the nucleic acid is there in the form of DNA. Now comes to the eukaryotic cell, chloroplast. Chloroplast is a double membrane organelle. Okay. And they also have its own DNA structure. It is having thylakoids that stacked over and form a grana structure, stroma lamellae attached to the another grana structure. Okay, like this. There are a number of structures like this, grana and stroma lamellae, and the matrix is there, which is called as stroma. Now, in this stroma, this DNA is present. A part of DNA, ribosomes are also present. Mitochondria, they are also the double membrane organelle. Its inner membrane invaginate and form crystal-like structure like this. They are also having a matrix, a substance which is called as matrix, and inside this matrix, DNA is present. Ribosomes are also present here. So we can see the DNA is present in nucleus, it is present in chloroplast, and it is also present in mitochondria. Now, if we talk about the prokaryotic cell, then apart from this main DNA, okay, there are some circular DNA are also present, which is called as plasmids. Okay, so this DNA is present in prokaryote and eukaryote in different organelles. Now, what are the features of DNA? So, if we are going to study about the DNA, we will study this DNA structure in these different headings. Nucleotides, how the nucleotides are formed. Polarity, DNA is having a certain polarity. 3' to 5' phosphodiester bond. So there is a bond which forms the backbone of the DNA, that is phosphodiester bond. Complementary base pairing, this is very, very important. How the two different strands of DNA join together. They are joined together with the help of nitrogenous bases. Now, how these nitrogenous bases are having a bond in between them. That is complementary base pairing. Double helical structure. How this helix structure exists. This we are going to study. Chargaff's rule. This is very, very important from the examination point of view. That also gives you some estimate of the percentage of nitrogenous bases present. The nitrogenous bases are present in the two forms, purine and pyrimidine. So in detail, we are going to study about all the features of DNA in the coming slides. So the first one, nitrogenous bases in DNA. As we told, purines and pyrimidines, two different types of nitrogenous bases are there. Single circular, or we can say single cycle nitrogenous bases, pyrimidines. Okay, that is cytosine and thymine. Thymine is only present in DNA. When it is RNA, instead of thymine, uracil will be there. But here we are going to study DNA, then cytosine and thymine. These are the two different types of pyrimidine structure. Now here you can see what is the basic difference between the pyrimidines and purines. It is a two-ring structure. Purines are two-ring structure. And the examples of purines are two, adenine and guanine. Cytosine is represented by the first letter C. Thymine is represented by the first letter T. Adenine is represented by A and guanine is represented by G. So these are the two different nitrogenous bases present in the DNA. Complementary base pairing refers to the specific pairing of the nitrogenous bases in the DNA and RNA. So how they base pair? Now, their base pair is very, very specific. Adenine always pair with thymine with the help of two double bonds. Cytosine always pair with guanine with the help of three hydrogen bonds. Adenine never pair with cytosine or guanine. It always pair with the thymine. And same way, thymine always pair with the adenine, never pair with cytosine or guanine. If we talk about the RNA, then adenine always base pair with uracil. It pairs with uracil. Again, it will be two double bond present between A and U. Cytosine, it always pairs with guanine, or we can say guanine always pair with cytosine with three hydrogen bonds. So you must remember that in DNA, thymine is present, and in RNA, instead of thymine, uracil is present. Rest three nitrogenous bases are common. Now, the nucleosides and nucleotides. There is a lot of confusion between these two structures, nucleosides and nucleotide. When the nucleoside is formed and when nucleotide is formed. So, nucleosides and nucleotides are the fundamental component of the nucleic acid. So, before nucleic acid is formed, first it has to form a nucleoside structure, then it has to form a nucleotide structure, and then it forms a helical chain-like structure. So they play a critical role in DNA replication, RNA synthesis, and also protein synthesis. So these two structures are very, very important. That is why it is called as fundamental components of the nucleic acids. So nucleoside is composed of two main components. Remember, only two main components, nitrogenous bases and pentose sugar, or we can just remember with the nitrogenous base and sugar. Okay, so nucleoside is formed with the help of two main components, nitrogenous base and pentose sugar. Nucleosides do not contain phosphate group. Always remember this. Sometimes the difference is asked, and sometimes the MCQs is totally based upon this. Okay, in which of the following structure phosphate group is present? So it is present in nucleotide. Phosphate is never present in nucleosides. Okay, so nucleotide is composed of three components: nitrogenous base, pentose sugar, and phosphate group. So if you see in the figure, sugar molecule and nitrogenous base, when these two are joined together, it forms nucleoside structure. Now this nucleoside, when joined with the phosphate group, it forms nucleotide structure. It forms nucleotide structure. That is why it is made up of three components, and nucleoside is made up of just two components: sugar and nitrogenous base. Nucleotides are the building block of the nucleic acid. So this is the building block of nucleic acid. They link together through the phosphate group to form the sugar phosphate backbone of DNA. So this is very, very important. Sugar phosphate backbone. So these bonds are formed and form a chain-like structure. So this is, you can see a chain-like structure, how this is formed. Now the DNA is having polarity. DNA polarity refers to the directionality or orientation of the DNA molecule. They are always having an orientation. Now if you see, one of the strand is having 5 prime and is here, then it will have 3 prime end here. Another end, 3 prime end is here, then it will end with 5 prime this side. So this is showing some polar structure or polarity. The DNA consists of two long chain or strands of the nucleotides that run parallel to each other. So they are running parallel to each other. This is one strand and this is another strand. Both are parallel and helically twisted. So one strand run in the 5 prime to 3 prime direction, while the other run 3 prime to 5 prime direction. That is why it is having polarity. Now when they form a polarity, some grooves like structure, you can see here the space here, the space here. Here the space is larger, then it is called as major groove. When there is the space is left, then it is space is small or less, then it is called as minor groove. Okay, so this is the polarity in DNA. Types of bond involved in DNA. Phosphodiester bond. These are the strong covalent bond that link the individual nucleotide together to form the sugar phosphate backbone of the DNA strand. So here you can see this bond and this particular bond. Phosphodiester, 2 ester bond is there, then it is called as phosphodiester linkage. And this ultimately forms the backbone of the DNA. Glycosidic bonds. N-glycosidic bond involved in the attachment of nitrogen atom, carbon of the sugar molecule. So here you can see, this is the sugar molecule. This carbon atom is attached with the one of the carbon of the glycosidic bond, or nitrogen of the glycosidic, nitrogen of the nitrogenous base structure. This is the base structure, and this particular bond is called as glycosidic bond. Complementary base pairing is crucial for the structure of replication of DNA. So this is very important. You can see here, adenine pairs with thymine, adenine pair with thymine, thymine pair with adenine, thymine pair with adenine. So never you will find that adenine is pairing with cytosine or adenine is pairing with guanine. You will never find. So adenine always pair with thymine or thymine always pair with adenine. This is fixed. Okay, and the formation of pair is with the help of 2 hydrogen bonds. Now comes to the GC. G always pair with C and C always pair with G with the help of 3 hydrogen bonds. Okay. Double helical structure was first elucidated by the James Watson and Francis Crick. So Watson and Crick, you will always remember with this name that the double helical structure was elucidated by or given by Watson and Crick in 1953. So DNA consists of 2 long chain, also called as strand, made up of nucleic acid. So this is the helical structure, one strand, and this is the another strand. They are parallel to each other, but they are helically twisted. The 2 DNA strands run in opposite direction, which means that they are oriented in an anti-parallel fashion. So if this is 3 prime, then this end will be 5 prime. And if this is 5 prime, then this end will be 3 prime. So this is anti-parallel fashion, and with this help of this only we can say that DNA is having polarity. The nitrogenous base of the 2 DNA strand form the hydrogen bond with each other. Adenine pair with thymine and cytosine pair with guanine. This creates a spiral structure that resembles a twisted ladder. This structure is often referred to as double helical structure. You will always find written as double helical structure of DNA. Okay, so 2 helix are there. That is why it is called as double helical structure. Okay, now the Chargaff rules. This rule allows a determine that the percentage of all bases present in DNA sample if we know that the percentage of any one basis that is present. So if you know any nitrogenous base percentage, suppose adenine in a sample of a DNA is having 20%, then you can find out what is the percentage of thymine, what is the percentage of cytosine, and what is the percentage of guanine. So interesting, na? So this is Chargaff's rule. So according to this rule, percentage of purine is always equal to pyrimidine. So let us understand this with the help of one example. Suppose in a DNA sample, adenine percentage is 30%. Then what will be the percentage of other nitrogenous bases? So we know that adenine is always pair with thymine, and how much adenine is there, that much thymine will also be there. So percentage of adenine is always equal to the thymine. So if A is 30%, then T will also be 30%. Then what is the total percentage of A and T? 30 plus 30. You will get 60%. So in that DNA sample, the rest out of 100 will be what? G and C. So total we are having A and T 60%. Then what is the left? 40%. So G plus C means 100% minus 60%, that is 40% is having G plus C. And G is again also equal to C as A was equal to T. So the total percentage of G and C is 40% and they are equally present. So 40 divided by 2, 20%. G is there and G is equal to C. So C will also be 20%. So with the help of this rule, you can find out the percentage of nitrogenous bases present in a sample. Okay, now we studied about the DNA. The another nucleic acid is RNA, which is also called as ribose nucleic acid. Now the ribose nucleic acid are of 3 different types. The first one is ribosomal RNA, and it is written as rRNA. R is small, rRNA. R stand for ribosomes. It is the most abundant RNA. 70 to 80% of the total RNA is ribosomal RNA. RNAs of eukaryotes. Now there is a difference. Eukaryotic ribosomes and prokaryotic ribosomes. Both are different. Okay. So eukaryotes, they are having 4 different types of ribosomal RNA. 28S, 18S, 5.8S and 5S. Prokaryotic ribosomes, they are 3 types. 23S, 16S and 5S. S for sedimentation coefficient. So here in the structure also you can see the prokaryotic ribosomes is 70S type and eukaryotic is having 80S type. And both, whether it is a prokaryotes RNA or eukaryotic RNA, they are made up of 2 subunits. But their subunits are not same. If you see in the case of eukaryotic, 60S is the larger subunit and 40S is the smaller subunit. Now comes to the prokaryote. 50S is the larger subunit and 30S is the smaller subunit. That is why 70S and 80S type of ribosomes is present in prokaryotes and eukaryote respectively. Now if you see the type of rRNA present in eukaryotic, the larger subunit, it is 3 types: 28S, 5.8S and 5S. Along with this, 40 different proteins are also present. Comes to the smaller subunit of eukaryotic rRNA. 18S rRNA is present and 30 different proteins are also there. Comes to the prokaryotic larger subunit. 23S and 5S rRNA and 34 proteins. Smaller subunit, 16S rRNA and 21 proteins. So there are lot of difference between the prokaryotic rRNA and eukaryotic rRNA. So 28S, 5.8S and 5S occur in the larger subunit of ribosomes, while 18S found in the smaller subunit of ribosomes in the case of eukaryotes. rRNA is transcribed in the form of longer chain of 45S in case of eukaryotes and 30S in the prokaryotes. After the transcription, this is such a longer chain and later it gets divided. It is broken into smaller subunits. 30S subunits of prokaryote ribosome has 16S rRNA and 21 protein molecules. 50S subunits of prokaryotic ribosome, 23S and 5S rRNA and some 34 protein molecules are present. Coming to the eukaryotic one, 60S subunit, it contains 28S rRNA, 5S rRNA and 5.8S rRNA and about 40 different protein molecules are present. 40S subunits of eukaryote ribosome consist 18S single rRNA type, 18S rRNA and some 30 proteins. Now comes to the third type of RNA, transfer RNA. This is the smallest one and this is also called as the soluble or sRNA. There are over 100 types of transfer RNAs and this is only the 15% of the total RNA present. So transfer RNA constitutes about 15% of the total RNA. So major one is the ribosomal RNA. Transfer RNA is the smallest RNA and it is made up of just 70 to 85 nucleotides with the sedimentation coefficient of 4S. If you remember, in the ribosomal RNA, the smallest one was 5s. Now this tRNA is more smaller than that subunit, that is 4s. 5 regions are unpaired or a single stranded amino acid binding sites. So we are having 5 different regions in the tRNA molecule. T5C loop. So here you can see T5C loop like structure. DHU loop. This is the DHU loop like structure. These loop is formed because there is no hydrogen bonding present. So they form a loop like structure. Okay. Extra arm. Here you can see this extra arm or variable arm. This is not fixed. That is why it is called as extra arm or variable arm. Anticodon loop. This is very, very important. Anticodon loop. So here, this is the mRNA molecule, and this anticodon loop is having 3 codons. 1 codon made up of 3 different amino acids or nitrogenous base, you can see. Now this codon is very specific to the attached amino acid here. Okay, acceptor arm. So this is the acceptor arm. So this is the 3-dimensional structure of the tRNA, and this is the 2-dimensional structure in the tRNA. So these are the different structural modification we can see in the case of tRNA molecule. Now every loop or every arm is having a specific function. Let us see what are those. Anticodon loop. It is made up of 3 nitrogenous bases. 3 nitrogenous bases. Here you can see CUG, 3 nitrogenous bases. For recognizing and attaching to the codon of mRNA. So according to the codon, this anticodon is there. And this anticodon is having a respective amino acid attached to it. Okay. Acceptor arm or the AA binding site. Now it lies at the 3' end. Now this is the 3' end of this particular tRNA. Opposite to the anticodon. This is the anticodon. The AA acceptor arm or the AA binding site is just opposite to the anticodon loop. And this is having CCAOH group. CCAOH group, to which the coming amino acid get attached. So the CCA group is added after the transcription. So this is the modification. This is not permanent or this is not a transcribed form. Okay, this is added after the transcription, means this is the modification of the tRNA structure. Amino acid or AA binding site and anticodon are the 2 recognition sites of tRNA. So this is recognized and this is the one which according to the codon present on the mRNA. Okay. T5C loop. It contains pseudouridine. It is the modification of uridine. That is why it is called as pseudouridine. This loop is the site of attaching to the ribosome. So T5C loop, with the help of this loop, it get attached to the ribosome. So every loop is having some specific function. Extra arm. It is variable site arm or the loop which lies between the T5C and anticodon. So it is present between the T5C and anticodon. This variable arm, which is also called as an extra arm. The exact role of extra arm till date is not known. DHU loop. This loop contains the dihydrouridine. This is again modification of the uridine. It is the binding site of amino acyl synthetase enzyme. This is very, very important. So DHU arm is important as it is the binding site of the amino acyl synthetase enzyme. So tRNA carries specific amino acid at a particular point during the polypeptide synthesis as per the codon of mRNA. So the codon which is present on the mRNA is read by the particular tRNA which is having a particular amino acid attached to it. So that particular amino acid attached tRNA comes and joined to that particular site mRNA that is having that codon. Now, codon are recognized by the anticodons of tRNA. This only just now I explained that codons are read by the anticodons of the tRNA. Now, the specific amino acids are recognized by the particular activating amino acid synthetase enzyme, and that get attached to it. Now, the third one is messenger RNA, which is also called as mRNA. It is a long RNA which constitute 2 to 5% of the total RNA. So, this is the least type of RNA present in the cytoplasm. 2 to 5% only. It brings instructions from the DNA for the formation of particular type of polypeptide. So if you remember central dogma of the molecular biology, so this is DNA. Now from the DNA, again DNA is formed. This process is called as replication. This is called as replication. Now from DNA, RNA is formed. This is called transcription. From RNA, protein is formed, and this is called translation. Okay, so from DNA, instruction is given to which molecule? RNA. Now the mRNA which is formed, it get modified. It is having at 5' end capping, so that some extra nucleotide get attached to it, and that is called as capping. 5' end will not get destroyed with the help of nuclease enzyme, because that enzyme is present there in the nucleus. So it can destroy this mRNA. So it get modified at this particular end, and it protect the 5' end. Similarly, a poly A tail is get attached to the 3' end, and it protect the 3' end. So here is the start codon and here is the stop codon. These ends are having some extra nucleotide which provide protection to the mRNA. So the instructions are present in the base sequences of its nucleotides. It is called as genetic code. So nucleotides which are present in the mRNA came from the DNA molecule by the process of transcription. So now it is having all the genetic code on it. 3 adjacent nitrogenous base specify a particular amino acid. Now 3 nitrogenous bases code for one particular amino acid. So 3,3 codon is read, and that particular amino acid get attached to the mRNA molecule with the help of tRNA. So formation of polypeptide occur over the ribosome, and here ribosome involvement is also there. So now here you can see, for the formation of protein, mRNA is required, tRNA is required, and ribosome, that is having rRNA, that is also required. So this is all about the nucleic acid. We will meet one more session of biomolecules. Till then, thank you very much.